Hello, everybody. Our world is so much diverse that all new discoveries year by year bring us new sensations about our existence. And today we're going to talk with highly prominent person, with whom I believe everyone could talk and find mutual points and talk long hours every single day. In a row of multiple outstanding awards, Professor Wayne Schekman, 2013 Nobel Laureate in Physiology or Medicine. Professor Schekman, thank you very much for joining us. It's a great honor, and I really appreciate this.
Sure.
So,
happy to be with you, Alex.
Thank you very much. Okay, the first thing I want to know, because I'm really curious of the thing about the development of the talents since childhood. So have you always wanted to be a biologist?
Not always. But my interest developed very young. I was probably 12 or 13 when I first became interested in biology. I think I was interested in science before then, but it focused on biology when I got a toy microscope. I collected some dirty water from a pond near my home in Southern California, and I put it on a glass slide, and I saw this world of creatures swimming and crawling around. It was quite remarkable, and I was captivated by it. And I've been interested in microorganisms ever since then. So every year after that, I did an independent project for a science fair that was held at the school. And in high school, every year I went to the California State High School Science Fair, and it was a good feeling. I enjoyed it, and I felt that was my niche. That was something I was certainly very different than anybody in my high school. And then I went to UCLA, which was relatively near my home. And I had an opportunity to work in a research laboratory in my first year. And that was really special because the professor with whom I worked just briefly was studying bacteria and bacterial viruses. And he had me read a book that was the first edition of the book by James Watson called The Molecular Biology of the Gene. And to me, that was an amazing revelation to read about this in a way that I hadn't read before. I'd read other science books, other microbiology books, but this book was quite unique in the way that Watson treated the subject, and it made it very exciting to me. And I realized through that this is what I wanted to do with my life. I had started college intending to be a physician, but that quickly evaporated when I realized that really what I wanted to be was a scientist. And so from then on, I became involved in working in research laboratories and was able to publish some papers while I was a university student. And then my path was clear.
That's amazing. Great. And about your research laboratory, what does it do now?
What is my research now? Yeah.
What are you doing with the team of the research laboratory now?
Yeah, no. Well, so in my independent career, I've always worked on membrane assembly, vesicle traffic. For most of that time, I worked on yeast, baker's yeast. But some years ago, we switched to working on cultured cells. Now we work on human cells in culture. And we're focused on the production and export of small, what are called extracellular vesicles or exosomes, and how they acquire proteins and RNA and what they may do if they're delivered to a target cell. So it's still kind of a biochemical approach to understanding how membranes function, how they're put together and how they function.
Okay, great. And how can your discovery of 2015 be applied in real life as per 2020 current developments, like in science, technology, and medicine as well?
Yeah. Well, I'm a basic scientist, so I'm not doing anything immediate of practical value, but I do work with biotech companies to try to implement some of the things that we've done in the lab. And now with these extracellular vesicles, there's a lot of interest in using these things diagnostically, because these extracellular vesicles, for instance, are present in our blood. And there's some evidence that the molecules that they carry, particularly small RNA molecules, may change during metastatic cancer. So, and if that's true, then they become a possible diagnostic tool. In a, you have an annual blood test, and normally they measure small molecules and cholesterol and things like that. Well, they may be able to measure the small RNAs in your blood, and that may be have a kind of a signature of changes that may be occurring, including metastatic cancer, and that would be really valuable. So that's one. The other thing is these vesicles may be part of metastatic cancer. There's some people who believe that these things target other tissues and cause a primary tumor to be able to migrate to another tissue to form a metastatic tumor. And if that's true, then there's maybe some way to block the production of these vesicles would be possible therapy, some kind of maybe a chemical or immune therapy that would be different than others. And finally, it may be that these little vesicles can be used to deliver drugs to a target cell. So there are many opportunities that people are exploring in various biotech companies. And so I'm very involved in helping a few companies try to realize this.
Okay, that's really good. Thank you. The book that got inspired and influenced you the most, or it was like that you told about James Watson?
Yeah, well, it was Watson's, it was his two books. First was the textbook, Molecular Biology of the Gene. That was what really got me going. And then his sort of autobiography called The Double Helix. That was, at the time, it was kind of scandalous. I don't know if you know that book, but it was published in 1967, I believe. And it was pretty scandalous because he was rather brutal with other people. He's always been very blunt, and he didn't, he wasn't exactly kind to some people. So anyway, of course, I didn't recognize that myself because I didn't know these people, but.
We're talking about the book, but not the people, so.
Yeah. So I enjoyed the book. I think it captured the flavor of what was, the most important discovery in biology in the 20th century. So, it's a great story.
Okay, great, thank you. But in this case, as you told about the double helix, I got another question. Like, how do you think there is kind of information that DNA molecule has not one pair of helices, but as many as 6 pairs? Do you think it can be true? And if yes or not, in any case, it is possible to assert that in our world there could be such phenomena that human beings have not reached yet in their own discoveries, or they are not ready to come to the perception of such kind of information, even if it could be a real discovery?
Well, you're asking two very different questions. The first is multiple helices. I'm not sure what you're talking about there. I'm not aware of up to 6 helices. What you mean that a single double
There's
strand?
kind of information that there is not like one pair of helices, but 6 pairs. So do you think it could be true or not? Like,
well, I mean, in our chromosome, in our cells, we have two copies of each gene. We're diploid. There are some organisms that are polyploid that have more than 6 copies of a gene, but I'm not aware, there are some genes, some regions of the chromosome that replicate many times over and then are held separate. They're, mini chromosomes that are transmitted. that may have many copies of a gene that has been amplified. But I'm not sure I know what you're referring to when you say 6 helices. You mean one double strand that some has bubbles that have 6 helices?
No, but it's 12 helices as 6 pairs.
6 pairs. Okay, I'm not aware of the specific thing you're referring to.
Okay, I just mean that like if it's not true or just true, that if people can understand some kind of information, or they are just not ready for understanding of some phenomena in our world. So
yeah, well, there are many things that we have yet to discover. That's for sure. We're very far from understanding everything. That, you know, the human brain is still a mystery. There are maybe 10 billion nerve cells in our brain, and each one may have 1,000 connections. So, you know, you could have over 1 trillion synaptic connections in the brain. So that's a very powerful computer. And, you know, I think it'll be many, many years before we have a deep understanding of that.
Yeah, we are on the verge of brand new discoveries.
Oh yeah, yeah, always.
Okay, concerning the world of publications, you are a supporter of the open access system. Mm-hmm. And why do you think for a journal which brings the real value that this kind of sponsoring model is more favorable than the paid subscription or just buying the separate article?
Yeah, well, you know, you've seen during the pandemic that all the journals, even the clinical journals, have made their papers relevant to COVID open access. They've done that because of the general public interest in that, and it's been amazing how quickly the literature is spread around the world as a result of that. And so that's an obvious, enormous benefit. And yet these same journals, these commercial journals, have not done the same for their other papers. You can only read the COVID papers or SARS-CoV-2 papers. You can't read the other papers that they publish openly, unless you have a license. So my feeling is that, you know, most of the research that we do around the world is paid by the public. It's paid through taxes, and yet the literature is held behind a paywall. So much so that even the author, who author of the paper, you know, has to be an institution that has a license and pays fees.
Yeah.
I think there are enough interested people out there, physicians, startup biotech companies, who don't have access to the relevant literature that should. You know, they're paying taxes, they should have access. And it's just, you know, it's largely because of these very, very big commercial outfits like Springer Nature or Elsevier that enjoy tremendous profit. They want to keep control because it's, you know, it's their profit. And I don't think we need them, frankly. I think we can, you know, I think active scholars can manage their own affairs. We don't need professional editors or these commercial publishers to broadcast our work. We can, you know, I started an open access online journal, eLife. We had support from private foundations, and the journal did very well, continues to grow. And we recover much of what we need through open access fees that the author pays. And eventually the journal will be self-sustaining without having to have a big profit margin. There are plenty of journals that don't make a lot of money that do a good job. So I don't see why we need to have commercial control. So I really am opposed to these big commercial publishers. I was also editor of the Journal of the National Academy of Sciences, the PNAS. And the PNAS by charter can make no money. They're not allowed to make any money, and they're not allowed to be a burden to the National Academy. They have to break even, and they do a fine job, and I think they'll manage to find a way to get enough income from open access fees.
Okay, and about eLife journal, I actually made the deep analysis, and it's really remarkable. I think so. But as a founding editor-in-chief, how do you think what the main challenges that have been faced in the journal, and yeah, the main challenges?
The main challenge is always the competition with the very so-called high-profile journals. You know, these commercially very successful journals, Nature, in the life sciences, Nature, Cell, and Science. These are very powerful journals. The journals have a high-profile brand name, so scholars feel, you know, that they won't make a career successfully unless they publish in these journals. It's just become so embedded in the culture that you have to publish in these high-profile journals. And you know, frankly, I don't accept that. I don't see why. You know, I published in these journals quite a bit when I was younger, but now I look back and I see, you know, I'm grateful that I was able to publish these things and it made an impact, but I don't see the need to have supported these large commercial ventures. You know, the guy who started the journal Cell, Benjamin Lewin, did a great job, you know, made a very popular journal. But, you know, the way he did it was by preying on the vanity of scientists. You know, so suddenly you wanted to be a part of this super exclusive club, and the entrance fee was very high, not in terms of money, but in terms of really hard work. And you know, he was very selective, and he had his own taste about what he wanted to publish. And then when he decided to retire, he was able to sell his business for $100 million. He made a huge profit. I mean, and it was sold to Elsevier, and they're continuing to make a huge profit. You know, and scientists just willingly pay just for the, you know, to have a star next to their name. And I don't see the need. I don't see the need.
Yeah.
Anyway,
yeah.
So I've stepped down as the editor of eLife. I'm on to other things now, but I still have the same attitude about this. And I've been pushing through this European effort called Coalition S for Plan S. Have you heard about Plan S? Many European organizations have come together to support a push for all the publishers to have open access. And that is having a big influence. Just this week, the Howard Hughes Medical Institute, I'm an investigator. That's a very big, powerful private funder in the US. Finally, after much deliberation, they've decided that all of their investigators will be obliged to publish all of their papers in open access journals. And that has really put a lot of pressure on Nature and Cell and Science to have some kind of open access policy. So I think that's good. I think that's very good.
Yeah, and that's a good time to make disruptive steps in this system.
Yeah.
Okay, and what were the most common reasons for refusal in publication in eLife journal?
What are the most common reasons? Well, the journal is selective. I think their acceptance rate now is about 20%, something like that. So it's tough, but it's not ridiculous. And usually, before a paper is committed for review, the work has to be judged to be, you know, different and new and interesting in the area of research where the work has been conducted. And then, you know, if it's considered interesting and new for the area, then it's committed to full review. And at that point, the chances are very good that the paper will eventually be published. There may be some revisions that are necessary, but I think the major reason that a paper doesn't get considered in the initial thing is it's just repetitious of something else that's already been done, or it's not not a significant development, sort of maybe more incremental.
I understand. Let's move a bit aside. The weirdest project you have ever taken part in?
The weirdest?
The weirdest project you have ever taken part in?
You mean in my research?
Yeah, in all your career and activities.
No, all my activities.
Very curious about.
Yeah, weird. Well, no one's ever asked me that. Weird.
Just into your opinion, what it could be.
Yeah, I'm trying to think what I'm pretty conventional. I'm not sure I've done anything terribly weird. Well, I mean, I have a mischievous sense of humor. So in my department here at Berkeley, we have annual retreats where the whole department goes and we hear talks. And I would always play practical jokes on people. And when I would lead a session, I would very often come out dressed up in a costume. And one year I dressed up as the resurrected Howard Hughes. You know, Howard Hughes was a very famous but reclusive industrialist. And when before he died, he became like a ghost figure living in a hotel room. He was very fearful of bugs, and he had, you know, a long beard, and he looked bedraggled, and he had long fingernails. And so I dressed myself up in a hospital gown and put on long fingernails and had a mask and long hair, and I wheeled myself in a wheelchair out onto the stage in front of the audience. And they were kind of, I think they knew it was me because I did this sort of thing, but I think they were kind of shocked. And my students were very embarrassed. So
that's amazing and really unusual thing. It's very interesting to know.
Yeah.
Great. Thank you for sharing this.
Yeah, sure.
Okay. So I also want to ask you about the role of wife in achieving some success in careers, work, new discoveries.
The role of my wife.
The role of a wife,
yeah,
just in achieving success in a work, career, discoveries, both like global and personal. Yeah. How great is it, into your opinion?
So I think I heard you right. This is the role. So my wife. My wife or my life? I didn't hear the word.
Wife. Yeah,
wife. W-I-F-E. Wife. Okay. Yeah.
Yes.
So I was married when I was in graduate school, and my wife was a nurse, and we had a great life together. We raised a family with 2 children who were grown up now, and then in around 1996, she developed Parkinson's disease. And that changed things slowly over time. And she became increasingly ill. She had to stop working. She developed dementia. And she died. 3 years ago, she died. So it was a wonderful life. She was very supportive. She was not as driven by her career as I was. So, you know, I helped around at the house, but it was mainly her. She took time off when the children were little, but that was her choice. And then near the end, in the last few years, as it got worse, I had to take care of her. And it became increasingly difficult because she couldn't talk. She could still walk, but her short-term memory was very bad. I'm on an automatic light that goes off when I'm not moving. Yeah, so we had a great life together, and now she's gone. But you know, I often reflect on how fortunate I was that I was able to have such a supportive wife. That was a benefit to my career. Now I have a new partner. She's a professor. she was a professor at UCLA, but she's moved up here and she's now a professor in my department. She's very different. She's very driven by her own career. But at this point, that's fine. I can take a back seat to her. So it's a very different sort of relationship, but it's equally supportive. So yeah, I mean, I think whatever, you know, having a partner or spouse, male or female, is a great thing. You know, I can't imagine having gone through a life without that. I knew when I was very young that I would want to have a companion. So I'm fortunate to have had that.
Great, thank you. All right, next question. I got the thing that you sponsored all the Nobel Prize funds to the establishment of the Esther and Wayne Schekman chair in basic cancer biology at UC Berkeley, named after your mother and sister.
Right.
For me, actually, it's also a personal thing because 3 of my relatives died from cancer. Yeah. And have you ever heard or watched the documentary TV series, which is called The Truth About Cancer: Quest for the Cures by Ty Bollinger? This is a documentary with the participation of 28 doctors, 11 scientists, 9 survivors, and 1 FDA-slain attorney who break the code of silence and expose the truth about the cancer and how we can actually prevent, treat, and beat it 100% naturally.
Well, I've not seen that program, and although I'm optimistic that we will be able to cure most cancers, it's not yet possible to prevent it.
Yeah, but the main thing I want to ask you, do you think that modern science, modern scientists can help to find out what cancer really is, and to elaborate the common accepted methods in curing cancer without surgery, without radio or chemotherapies? How do you think would it be possible?
Well, you know, I mean, I believe in the scientific method, and I don't believe in unproven theories. And the evidence is pretty substantial that home remedies and incantations and prayers do nothing. So I believe in medical intervention, and there have been great strides. A former colleague of mine who was at Berkeley and is now in Houston, James Allison, won the Nobel Prize for his development of immunotherapy for cancer. And it's been a tremendous success for certain cancers. Certain cancers now that were a death sentence can be largely treated. But it's not all cancers, and so we need to find new ways. Cancer is not one disease. It's maybe dozens of different diseases. And we need to find treatments that are effective for the different forms of cancer. But in any case, these will all involve some kind of physical intervention, either you know, empowering the immune system, or drugs, or directed radiation. I don't believe that changing one's diet or exercise alone can do it. You will need to have intervention, possibly even genetic intervention. And so the trouble with these popular alternative theories is that there's no evidence to back them up. There's no proper clinical trial to show that these things have any effect at all. And so, you know, there are lots of people who've written popular books that claim that they can prevent cancer by the right diet, but the evidence does not back that up at all.
Okay.
So
I'm not implying on it.
Yeah.
Like that.
Well, you know, I mean, there are lots of quacks out there who make claims. You know, snake oil salesmen. make claims. You know, when stem cells became an important topic of research and it became possible to make embryonic stem cells in the laboratory, suddenly there were clinics popping up all over the world claiming that they would treat patients with stem cells. But, you know, they were all, mostly they're just fly-by-night quacks, you know, looking to make a quick dollar. And so one has to be very cautious about people who make these claims, because they're usually, they're never based on science.
Okay, thank you. All right, I understand. Also, I want to ask you the question about the reproducibility of the results and the publication of some author as a project. For the time being, there are more than 20,000 of retractions worldwide, officially recognized only, and its number is continuously growing. And also in 2016, there was a panel discussion with your participation where you gave an example of Dr. Berkeley from MGM, who had a talk with some author of the problematic study, where Dr. Berkeley told that he read carefully line by line and tried to make an experiment 50 times, but eventually failed to find any kind of results. And the author replied that he tried only 6 times, and only once he got some kind of result, which he put into the paper as the best suitable story for publication. So this kind of problematic, just in your opinion, what kind of pivot could be done so that scientists will not chase high ratings, but enjoy the process of participation in quality research, performing quality publications and new discoveries?
Well, there's never going to be an end to it. You know, there's some dishonest people, even successful scientists. Some of them are dishonest, and there's, you know, some of them will hide their dishonest work, and it, you know, you just have to be vigilant, constantly vigilant. And so I think that the journals who publish this work should own up to their mistakes and should be willing to publish retractions and publish conflicting data that challenges something that they published. I think that when evidence of manipulation or fraud is brought forward by someone who challenges something that's been published, there needs to be a different way to evaluate whether this really was misrepresentation. The way the system works now, when someone writes to me as a journal editor and says, you should look at this paper, it's the data's been fabricated. The journal is not in a position to investigate. They can look at the data and they can say, well, it does look like there may be something wrong here, but you know, they don't have the investigative team to do this. So what happens is that the charge of manipulation is sent to the home institution of the investigator who published the paper. And they can then conduct an investigation. They have faculty in the university, but they are in a conflict of interest. Now, some institutions will take that charge very seriously and investigate thoroughly. But I can tell you, I know of a lot of institutions that will simply cover up for their investigator, because it's harmful to them when their own professor is charged with fraud. And I know of very specific examples of major institutions that simply, you know, they try to attack the whistleblower rather than to properly prosecute the case. So this needs to be removed. This is a flaw in the system. This needs to be removed from the home institution. And there should be some panel or some authority. In the US, there is such an authority in the NIH for life science research. It's called the Office of Research Integrity, but it's so bureaucratic that nothing ever gets done. It's just a black hole. So I've argued that some outside authority, in this case in the US, it could be the National Academy of Sciences, which is a private organization. They should be empowered to lead such investigations, and then to have a representative that then goes to the home institution and supervises the investigation. But that hasn't happened. And so a lot of the bad papers that get challenged never get retracted because they come to an impasse. And I can tell you from my experience when I was editor of the PNAS, they would rarely retract a paper, even when the evidence was clear cut, because they were worried about getting sued by the home institution. So they're very risk averse, and so the system is it's a real problem. Anyway, journals should own up to their mistakes, and the major journals, these so-called high-profile journals, are very reluctant. They're very reluctant to retract papers. They occasionally do, but they're very reluctant. And they are equally reluctant to publish other papers that challenge the work that they have previously published. Here's the most famous example is a paper published in this medical journal called The Lancet. It was a paper by a physician named Andrew Wakefield. This was published 15 years ago or so, maybe 20 years ago. He claimed indirectly that vaccinations may be the source of autism.
Yeah.
You know about this. Yeah. So this has had an enormous chilling effect on the public acceptance of vaccination, which right now is terribly important, as you can imagine. And so, but on subsequent investigation, it was some of the data was manipulated, and this guy Wakefield was just a fraud. He was fired from his home institution, but he now goes around the world, bringing up these opposition groups, he's now become a politician. He goes around challenging vaccination programs around the world. So that paper took 12 years before The Lancet finally retracted the paper. 12 years. So now here's a new trend that I think is really very healthy that may cut papers like that out. And that is, they're increasingly now in the life sciences and even in clinical science, there's pressure to post your paper as a preprint on an archive, like the bioRxiv. In physics, this has been going on for nearly 40 years, Physics Archive, and it's just an accepted tradition. You post the paper when you're ready to submit it for publication. And when that happens, you have many people looking at the papers. And so there are many opportunities to find serious flaws in the data. Whereas in the traditional review process, when a journal solicits reviews from 3, maybe 4 people, it's more limited, even 3 or 4 people can't catch all the possible flaws. But when you post it on the Physics Archive or on the bioRxiv or now the medRxiv, it's there for everyone in the world to look at. And there are hundreds of people who look at these papers. And so you can find flaws. And so I suspect that this Wakefield paper would have been called out as fraudulent, or suspicious at least, if it had been posted on the archive before it was reviewed by the Lancet. And then it wouldn't have been published. And then, maybe a lot of the damage that he's caused would have been prevented. So this is a good trend. Just have wider openness and transparency is the greatest power to prevent this kind of fraud. And so I think that it'll be increasingly difficult to publish things that are really overtly fraudulent because there'll be too many people looking and scrutinizing the data.
Yeah, let's
And you've
hope
seen
that,
examples of this now during the pandemic. There have been a couple of papers that were posted on the archive that were challenged right away, and I suspect these papers never got published as a result.
yeah. There are actually also lots of publications which have been retracted on
Yeah,
because,
there have been, but I think it's the tip of the iceberg in terms of manipulation.
yeah, for sure. All right, and I also want to ask you about what advice can you give to young scientists, which are also a part of our big time community?
Yeah, well, science in the life sciences are changing. I think the opportunities are greater than ever. I'm very upbeat about prospects. The opportunities for discovery and for application are tremendous. And I'm seeing in my own laboratory that many of my really excellent students want to go into biotechnology because there are many, many more opportunities than in academic science. The academic science can only grow, at a slow rate as people like me retire. But biotech just keeps growing rapidly. And this pandemic, if anything, will stimulate interest in biotechnology. So, and I think it's a great career path for many people. Now, some people will want to continue to be independent and focus on very basic issues. That's good. Obviously, I did that. But this option of being in biotech didn't exist when I was a graduate student. Now it's very prominent, certainly here in the US, but also in Europe. So I think this is a good trend. We've learned so much now and the technologies have advanced so rapidly that therapeutic possibilities are much more available than they used to be.
Yeah.
So I think that's good for both basic science. There's a lot of money that goes into basic science. I think, again, the pandemic will stimulate a lot of money invested in the NIH. I mean, for this pandemic, look at the damage. Over 1 million people died now, and many more yet to come. Trillions of dollars in the economy lost. This could have been prevented. This whole pandemic, not only controlled, but it could have been prevented. And it's very simple. When we had the SARS epidemic 18 years ago, and MERS 9 years ago, SARS was a terrible disease. It had a very high mortality rate, but it was self-contained because you weren't infectious until you had symptoms of the disease. COVID is much worse because you can spread the disease asymptomatically. But when the SARS epidemic first struck in Hong Kong and then in Toronto and a few other cities, there was a flurry of interest in the virus, coronavirus, and for an initial attempt to make a vaccine. But then they were able to, public health measures were able to control and eventually eliminate the disease, essentially. And so the pharmaceutical industry no longer had any incentive to develop drugs. Chemical drugs to inhibit the virus. And, viruses like the coronaviruses, there are many different coronaviruses, and they're all immunologically distinct. They have different surface antigens. So a vaccine that would have worked on SARS almost certainly would not have worked on SARS-CoV-2. So no one has a universal coronavirus vaccine, just like there's no universal flu vaccine. But with chemical therapies, the viral genes that are necessary for viral replication are not so free to mutate to become resistant to drugs. And so I think it would be possible to find a drug or a cocktail of drugs that completely inactivates most coronaviruses, just like we have with HIV. There's never been a successful vaccine for HIV because its surface changes so rapidly during the infection process that you can't Your body simply cannot mount an immune reaction. But with a cocktail of drugs, you can wipe the virus out completely. And I think if there had been a concerted public-private effort with the foreknowledge that eventually there would be an outbreak of coronavirus that was really deadly, as we're now experiencing, if that work had gone on during the previous 18 years, we would have had a drug or a cocktail of drugs to completely eliminate the disease right away. So that's a tragic failure of insight and investment, which we simply cannot afford to do again. We cannot make that mistake. And I think now, as a result of this pandemic, we will have a cocktail of drugs to keep at least another coronavirus infection from spreading like this. And so there needs to be an investment in universities, through the government, and in the pharmaceutical industry to develop a more complete set of drugs to block infectious agents, bacterial and viral. Because because the alternative is what we're suffering now. So,
but do you think it could be a vaccine or just some?
Well, people have said that they're trying to make a pan vaccine that would block all coronaviruses, but I'm dubious. I mean, people have been trying for over 30 years to get a vaccine against HIV. So I'm dubious. But with drugs, you can have a cocktail of drugs, as with HIV, 3 different drugs targeting different core components of the viral replication machinery is, virtually 100% effective. And we could have had that for coronavirus now if the investment had been made. Because, the investment would have been billions and billions of dollars, but the cost of failure has been trillions of dollars.
That's bad. In any way, we should take a hope that the world will move to the right direction. And
well, that's not a given. Politically, I don't see the world moving in the right direction. Not at all. Politically, it's moving in the wrong direction. But I'm still hopeful. The news today is that Trump is coronavirus positive. He has the infection. So, yeah. I don't wish anybody ill, but this should be a wake-up call for him. That it's not fake, it's real.
Okay. Okay, Professor Schekman, thank you very much. It was an exciting conversation, talk, lots of useful and motivating information to hear from you. I do believe that all the legacy you performed, all the achievements will be highly important for your followers and those who will make new, important in our world discoveries. Yeah, thank you very much.
Okay.
It's a great honor to talk. Thank you.
Nice to chat with you. Have a good evening.